Good evening, everyone, and welcome to the Australian Academy of Technology and Engineering Victorian Division seminar series. I'd like to start by acknowledging the traditional owners of the land on which I'm based in Hawthorn, the Wurundjeri people of the Kulin Nation, and to pay my respects to their elders, past, present, and emerging. We have two fantastic speakers for you this evening, and the topic tonight is medical devices transforming lives. We have Professor Tony Burkitt and Professor Brendan Fafiani. I've just promoted you, Brendan. You've been an honorary professor now. And I'd like to encourage all of you in the audience to post your questions to the Q&A. Professor Sandra Kentish will be taking the questions later on. So our first speaker for this evening, Tony, is Professor Tony Burkitt, who's a Professor of Biosignals and Biosystems at the University of Melbourne. He was the Director of Bionic Vision Australia from 2010 to 2016, which led to the development of a prototype bionic eye, the successful implantation in 3 patients, and the establishment of the company Bionic Vision Technologies, BVT, with US 18 million in venture capital for the ongoing commercial and clinical development of the technology. In addition to his work on the bionic eye, Professor Burkitt's research encompasses computational neuroscience, brain-computer interfaces, neuroengineering, cochlear implant speech processing, and biosignal processing for epilepsy. Thank you so much for joining us this evening, Tony. I'll pass to you.
Good, thank you very much, Bronwyn, and it's a great pleasure to be able to present this evening. Let me first of all just bring up my screen. We'll see if we can share the presentation. Yeah, so it's a great privilege to be able to present this evening. I had the great privilege of leading a national consortium to develop and commercialize a bionic eye. This was a project that was funded by the Australian Research Council, and you can see down the bottom here the different partners in the members and partners in the research collaboration, involving the University of Melbourne, the University of New South Wales, the Centre for Eye Research Australia, and NICTA, who are both based in Melbourne, and the Bionics Institute, also, of course, based also in Melbourne. And with partners, the Royal Victorian Eye and Ear Hospital, the University of Western Sydney, and the National Vision Research Institute. The project had its genesis really back in 2007, and we spent a couple of years building together a research team, as well as building together a business case, and engaging with government to seek funding for this project. And it was eventually funded in 2010 through an Australian Research Centre special research initiative in bionic technology. And so that's why we have here the Australian Research Council, who are the funding bodies for this. So the goal of the project, a very clear goal, was to develop a retinal device to restore vision to the blind. Now, this is unusual for the ARC to have an explicit commercialization goal in addition to the research, the fundamental research that underpinned this. But before I talk about the bionic eye itself, it builds upon a long history of technology, which is actually crucial to really understanding how the bionic eye works. And this goes right back to the heart pacemaker, and many of you perhaps won't realize that, in fact, Australia was one of the leaders in the development of the heart pacemaker technology. And the company at the time, Telectronics, had developed really world-leading expertise in both the electronics for implantable devices and the hermetic encapsulation of these devices to keep the electronics safe, both in terms of the safety for the person, for the patient, and for the electronics to keep it functioning for the lifetime of the patient. Now, this technology was then crucial to the development of the cochlear implant, which was undertaken by the team of Professor Graham Clark at the University of Melbourne. And this picture actually shows the first prototype cochlear implant that was developed by Graham and his team back in 1978. And it was this device, which eventually became commercialized, actually changed dramatically in that time, to become the device that forms the basis for Cochlear Limited, which of course is one of the leading biotechnology companies in Australia and worldwide, and has something of the order of about 70% of the world market in cochlear implants. There are a couple of other companies internationally, but it's by far and away the world leader in this field. So Australia has this really strong history in medical bionics, and our project actually leveraged off this enormously. There's a huge pool of expertise within Australia in this quite specialized field that really places Australia at the forefront internationally. It also meant that we had, as part of our project, the remit to continue the training for young researchers and students, research students in this technology, to make sure that Australia remains at the forefront internationally of this field. Now, for those of you not so familiar with the cochlear implant, this is just a very quick diagram just showing how the cochlear implant works. So we're seeing here a cutaway view of the ear. The group of patients for whom this is suitable are those deaf patients who suffer from what's called nerve deafness. And that typically involves the death of the nerve cells or the hair cells in the inner ear. And you can see the inner ear is this shell-shaped structure here. The cochlear implant itself consists of a couple of different components. This is the external component. So there's a microphone here, batteries, and a lot of electronics, all miniaturized, goes into this component, which sits behind the ear and is connected to this transmitter that transmits the electrical signal through to the implanted components that we see here. So there's an inductive coil that picks up both the power and the signal that are sent from the external components. The signal is then decoded there's an encapsulated electronics compartment here. And so this is the hermetically encapsulated electronics. And that is decoded and then sent down to as you can see here, to the electrode array that's implanted into the inner ear. There's a close-up view of the electrode array. And the little bands you can see here are the 22 platinum electrodes that send out the stimulating pulses that are then picked up by the surviving nerve cells in the inner ear. And these are then sent through the nerve, which then takes it then to the auditory brainstem, and then of course up to the brain where it's interpreted as sound. So, having seen that basics of how the cochlear implant works, I'll take you through how the bionic eye actually builds upon that technology. So first of all, we'll talk about the group of patients for whom this is appropriate, who benefit from this technology. And to do this, we need to first of all describe to you how healthy vision works. So what you see here is a picture taken through the front of the eye. And we see here the central part of the retina called the macula. And you can see the different blood vessels that go off here. And these blood vessels exit the eye at the optic nerve head. So that's where both the blood vessels and the nerve actually go out through the eyeball. And that corresponds to the blind spot. And it's this very high density of the photoreceptors in the central part of the retina that give us this high acuity vision that we have, enabled us to see enormous amount of detail with normal vision. Now, if we do a cutaway through the retina, you can see the different layers of the eye here. First of all, with the retina, and inside that there's a series of blood vessels that supply the oxygen and take away all of the products, because the retina is very highly active. And then the external part is the sclera. This is the white of the eye. And if we do a blow up of the retina itself, the retina consists of a number of different layers of neurons. So the light comes in through the front of the eyeball, has to actually go through the different layers of the retina before reaching the rods and cones that are at the back of the retina here. Now the signal is then, the light is transformed into electrical signals that are then transferred to these other neurons in the retina here, that then exit the retina through what's called the retinal ganglion cells. And this forms the optic nerve that carries the signal up to the brain. Now, for people who have some sort of retinal degeneration, that typically involves the death of these photoreceptors, the rods and the cones here at the back of the retina. So they have disappeared, but in fact, the other nerve cells in the retina typically remain there. And these can now become the targets for the electrical stimulation that we'll be using with the bionic eye. Now, the two conditions that we'll be particularly concentrating on, the first of these is retinitis pigmentosa, or also called RP, and it's the leading cause of inherited blindness. It affects about 1.5 million people worldwide, and around about a quarter of these patients are legally blind. The way in which it works, and you can see this picture here again showing the view through the front of the eye at the retina, this fundus image. The areas of blackness here, of discoloration, correspond to damage of the photoreceptors. And what happens there is that people develop this tunnel vision. They end up with these islands of relatively normal vision, but in fact, they've lost all of the peripheral vision around that. And of course, in the later stages of the disease, they'll actually lose even these islands of vision and become totally blind. The other form of retinal degeneration is this one called age-related macular degeneration. And this affects roughly 1 in 1,000 people worldwide, but it particularly affects elderly people. That's people over the age of 65. And so as our population ages, we have more and more elderly people. This has become the major cause of severe vision loss in Western countries. So for a country like Australia, it costs more than $2.5 billion each year just for this form of retinal degeneration. And you can see that what it does to the retina, you can, again, looking through the central part of the retina here, we get this discoloration. And the white here corresponds to the buildup of lipids, and the darker spots here correspond to the death of the photoreceptors. And so what people experience is the loss of vision in the very central part, this macular area of the retina. So complete loss of vision in the central part, but quite often they have this lower grade peripheral vision that, at least in the early stages of the disease, still enables people to move around in their environment, but they completely lose the ability to do things like read print or to recognize faces. Now, so what does a bionic eye look like? So this shows just a diagrammatic view of how the system works. And you see here this behind the ear unit, we call this is the receiver part. And so there will be an external component that I'll talk about in a moment. But this is where the signal is received from the external components and passed along in the same way as we saw for the cochlear implant. The electrical signal is passed down to the implanted components, which are implanted surgically very close to the retina in the back of the eye. Those then stimulate those surviving nerve cells, and that signal is then sent through the nerves back to the visual processing centers of the brain. So the different components of how this bionic eye system works are shown on this diagram. You can see here at the front, there's a very small camera mounted on a set of glasses. And this will be the one that captures a live video stream of the external input. That signal is then sent down through this wire here to a small body-worn processor that's not shown in this diagram, but it's about the size of a mobile phone. And that's where all of the image processing takes place. The signal is transformed from an image into the type of electrical stimulation that we need for the retinal implant. So all of that information is fed back up through this pathway, then through to the transmitter, which sends that through using radio frequencies to the implanted components. Sorry, I've missed, I've been meaning to press this on to show you the implanted components then decode that signal and send it through the wire here that takes it into the electrodes, the implanted electrodes at the back of the eye. And then those implanted electrodes then stimulate the nerve cells, the surviving nerve cells in the retina, and then it's sent back to the vision processing centers of the brain. So what is it that people see with this type of electrical stimulation? It's been known for a very, very long time, long before there was any talk of retinal implants, that any type of stimulation of the retina, whether it's mechanical or electrical, can also evoke a visual percept. And we call this a phosphene. And this is basically the induced perception of light by means other than light entering the eye. And so electrical stimulation at just part of the retina can actually cause this sort of percept or of a phosphene in a particular part of the visual field. Now, to give you an idea of the what it is that somebody with a retinal implant will see, this diagram shows essentially what happens with different numbers of electrodes. I'll give you a schematic view of the amount of visual information that patients will receive with different numbers of electrodes that correspond then to, as it were, phosphenes, or in this case, the number of pixels of an image. And you can see down the bottom right-hand side a higher resolution version of this picture, just a street scene. And you see with a very small number of pixels or electrodes, you get very little information. And as you increase the number of electrodes, you get more and more information. The current resolution of the sort of devices that have been developed in the last couple of years have been around about this sort of level. So it's still a very coarse, so we're talking tens of electrodes, very coarse level of vision. What we're seeing here, of course, though, is a very static image. In fact, what patients do is that they move their eye in order to be able to scan a visual scene. And so they can actually pick up slightly more information than you would actually get the impression by just looking at a very static image. What we would like to be able to do, of course, is to eventually be able to provide patients with a much higher resolution view of the external world. So that's sort of where we would be aiming in the future with this technology, although we're only at the very early stages of the development of it at the moment. So in the development of any medical bionics device, the two crucial aspects in order for this to become an effective technology are, first of all, safety, and secondly, the efficacy, how well it works. So we spent a very large part of the project essentially showing that the device was safe. And you can see actually a close-up now here of the device itself. So this is the stimulating electrode array, and it's encapsulated in silicone. So silicone, this substrate here with the platinum electrodes in it, both platinum and silicone, we know are inherently safe from the work that we've done over many, many years with cochlear implants. But that work, of course, has been done in the context of the ear. So we have to actually show that, in fact, these materials and the shape of the materials are safe when we put them into the eye. The cochlear implant, of course, very, very mature technology, and it's been widely used. Many hundreds of people, hundreds of thousands of people worldwide who have had that technology. But it's taken a couple of decades to be able to apply this to people who are blind. And part of the reason for that is that the retina is a very, very delicate organ. So we're seeing here a close-up view. This is a cutaway view of the eyeball here. And this blow-up section here shows the retina on the left-hand side of this image. And you can actually see the different layers of the retina. And the space here is actually where the electrode array was positioned behind the retina during these studies. So this is a 3-month implantation in an animal model to check that the retina itself isn't damaged in any way by the presence and the activation of this device. So this device has been in this animal for 3 months and stimulating the retina. And we can see with images like this, that these layers, the functional layers of the retina here, have in no way been damaged. and that in fact there hasn't been any buildup of tissue around the device in what's called the choroid here, and then of course the sclera behind it. So it was these long-term studies that gave us the confidence that the technology that we were developing was very safe, and particularly had to be safe for the long term. I mean, these devices, the cochlear implant is implanted as young as possible for children who are diagnosed as deaf, for children as young as 18 months, and it stays in there for the lifetime of the child. In fact, those implants don't need to be changed or in any other way repositioned or removed. And that's what we're aiming for with the retinal implant as well, is that the device will be actually safe for the lifetime of the patient. So the second thing that we need to show is mentioned there, the biocompatibility of all the materials, which is absolutely crucial. The second thing is to show that in fact the device is efficacious, that it actually works and will evoke a visual percept. And in order to do this, the first study we did was a prototype study to be able to demonstrate that the electrical stimulation from the point in which we're actually implanting the electrode will actually reach the surviving nerve cells and evoke the sort of visual percepts that will be useful to the patients. So our first study involved the implantation of a device in 3 patients in Melbourne. And these are our 3 bionic eye patients. And these are really quite extraordinary individuals. We really have a great debt of gratitude for their altruism and their dedication and engagement with the project. They themselves knew that this was a prototype study, so the devices that were being implanted in these patients weren't permanent. They were never envisaged to be permanent. But these patients undertook this quite often because they themselves also had family members who were suffering from a similar condition. Retinitis pigmentosa is quite often an inherited form of retinal degeneration. So they had younger members of their wider family who were also suffering from this disease. But during this time, they of course became very much a part of the research team and dedicated at least 1 day per week where they came into the labs and worked with us, undergoing all sorts of testing and to be able to determine how well the implant was working and what new features we would need for a more permanent device. So this just shows here now, you recognize the actual, on the right-hand side here, the implant itself. So it had 24 channels. And in order to do the prototype study, we didn't implant any of the microchip electronics. So it actually had what's called a percutaneous connection. We're using the materials with which we're familiar. And I'll talk a little bit about what's called this suprachoroidal placement, the surgical placement. All 3 of these patients suffered from retinitis pigmentosa, and they only had bare light perception. They were at the very sort of extreme end of vision loss because we didn't want to put at jeopardy for any patient who actually had functional vision. And like I said, they were implanted for a period of 24 months, 2 years. And the result of the study was that all the patients perceived these phosphenes, and we were able to test the device out with a camera attached in what we call a semi-portable system. But just to give you a feel for where the device was implanted into the retina, the different layers that we saw before, it turns out that you can actually create a small space here in between the choroid and the sclera. This is called the suprachoroidal position. And so the electrode itself can go into this position here, which actually keeps it a little bit away from the surviving nerve cells in the retina and means that these are protected from any mechanical movement or anything that happens here with this suprachoroidal positioning. We developed with the surgeons a safe surgical approach. So there's a long sort of intensive development of this with the surgeons to ensure that we actually had a very safe way of implanting the device and that it would remain stable over long periods of time. Quite a large surgical team. We're very fortunate to be able to team up with the Royal Victorian R&D Hospital. That is also the centre where the first cochlear implant was done in Australia. So enormous depth of expertise there in this technology from which we've benefited. So this shows our first patient, Diane Ashworth, at the day in which the device was actually switched on. And you can see here, she actually got a testing device here. This is a device which will actually track where her eyes are moving in relation to the electrical stimulation that she's receiving. And this shows the testing booth, which was set up at the Bionics Institute, where all of that testing with the patients was undertaken. And we have over here a stimulator, and then a router which actually directs the current to the different electrodes, emergency disconnection here. And then this is one of our graduate students with the eye tracker on here, so we can actually see track the eye movements in relation to the electrical stimulation that the patient's receiving. And they can map out with their finger, there's a finger tracker here, which can map out for the patient what it is and where it is that they're actually seeing the stimulation, the different forms of stimulation that we give. The before surgery, this is a picture looking through the eye for one of our patients. And we can do, and this shows the picture after the surgery, and you can actually see the outline of the retinal implant here. This is, of course, the optic nerve head. And this is the central part of the retina, the macula. And each one of these little circles here represents one of the electrode arrays. So we can actually use these sort of pictures to be able to monitor after surgery exactly the placement of the electrode and to be sure that it's not moving. Even more than that, we can do what's called the optical coherence tomography to monitor the cross-section of the retinal positioning. And you can see the electrode array here between the outer layers, the sclera, and the retina here, which is slightly pushed forward, but you can still see here the layers of the retina, which are still exactly as they were previously. They haven't been affected in any way by the implantation. Now, it turns out that the measures of visual acuity that we typically use for people who have low vision really aren't terribly appropriate for people who have a retinal implant. So we needed to work with the clinical team to develop real-world functions or measures of their visual function. And we did this through a variety of sort of orientation mobility tasks, and by getting them to carry out activities in daily living. So it was really a whole new sort of testing protocol to actually work out what sort of functional vision they could actually use in everyday life, because this is actually what interests the patients most. We also could test out what we call a semi-portable device. It's called semi-portable because it actually consists of a backpack here, in which we have up here the camera, which is collecting the image, an eye tracker there, so we can actually monitor throughout the time where their eye's moving. The backpack contains all of the electronics that they need, and of course a battery pack here. And we could take them both in the laboratory sort of setting, but also out onto the street to see how they could actually operate really in a more natural environment. And we could do things like testing obstacle avoidance using this mobile system. And that proved really a wonderful way of being able to test just how well the whole system worked for the patients in more realistic real-world situations. This shows a view of some of the patient testing that we did. This is actually taken in the hospital testing situation, where we got them to do tasks like making coffee, being able to identify sort of objects. Here we have a number of coins and things, being able to identify things like objects on the wall, where doors were, where windows were, things like that they would need to do in everyday life. So I wanted to finish off by talking a little bit about the commercialization pathway, because this was an absolutely sort of crucial part of the whole development of the project. So as I mentioned earlier, the aim of the research program was to really provide this proof of concept that the electrical stimulation would actually be able to generate useful visual percepts for the patients. Now, because this is really set up to be on a commercialization pathway, we needed to make sure that what we were doing actually also satisfied all the regulatory pathway requirements. This is quite a significant burden to any research program. And so we had to make sure we had early engagement with the regulatory authorities and to be carrying out all of the testing that we did both in the preclinical program and in the clinical program in a way that would satisfy the requirements of the regulatory authorities. Safety, of course, was absolutely paramount. This has been built in right from the very start of the project. And we know from our experience with the cochlear implant that this is actually what underlies the commercial success of any medical bionics device. Cochlear Limited has an absolutely exemplary track record in terms of safety, and we've sought to emulate that in every way. And we've also made sure that we're engaging with the expert opinion leaders. So these are leading clinicians internationally to make sure that we were engaging them, finding out what they felt was required for the patients, and keeping them up to date with the latest developments. It's also an important part of building confidence in the broader community about how the devices work. In terms directly of the commercialization, we also established a commercial company fairly early on in the development of the program. And initially, this was really just a IP holding company to make sure that we captured all of the intellectual property. And we had a quite rigorous process for the management of the intellectual property, making absolutely sure that everything was captured in a way that would be entirely watertight and useful for a commercial entity to subsequently build a commercialization company upon this. This also, in the latter stages of the project, involved some quite strategic decisions that had to be made about which parts of the IP portfolio were really the most important, and whether there were any gaps in the international IP portfolio or IP strategic landscape that we could actually also gain new pieces of IP in. So that's also a really important part in the whole commercialization pathway that we set out on right from the very start. So where is it up to at the moment? So this is a picture of the device that actually is going forward in the current clinical trial that's being undertaken with 4 RP, retinitis pigmentosa patients, and being led by Bionic Vision Technologies. That's their logo that you see actually up here on the right-hand side. So BVT is the one who is leading this and taken over the studies now, this patient trial with this device here. So it has many more electrodes than the prototype device that we had. It also has fully implanted electronics, and it's a device that can actually last the lifetime of the patient. And this is, again, the same schematic that we saw earlier. So this is actually the device that would then be implanted that has been implanted in these patients and the subsequent patients. So what are the compelling advantages that this technology has over the competition? Because this is a very active field worldwide. There are a number of international competitors. So some of the advantages is that the surgical placement that we developed avoids damage to what is an already compromised retina. These are patients with severe vision loss, and so the retina has already been damaged to some extent. And the surgical technique that we've developed really avoids any further damage, and it doesn't block or damage any remaining vision. Moreover, the surgery is relatively straightforward for a retinal specialist to perform as an outpatient procedure. So this is really, it only takes a couple of hours for a retinal specialist to implant the device. So that's a very important consideration in terms of the safety and management of these patients. We also have advanced software and hardware components that can be easily updated. All of these are external, and they can all be updated with the latest software as time goes on. Another important component is that the placement of the electrode array means that it doesn't preclude these patients from participating in any subsequent gene therapy studies that were to happen. If new technologies are developed over time, these patients could actually still benefit from alternative technologies that are quite different to the bionic eye that they're receiving through this device. So Bionic Vision Technologies has recently completed this initial study of now 7 patients and has really demonstrated both the safety and efficacy and the positive patient outcomes, showing that you can in fact improve patient mobility, and they also show that the device is stable and durable. They're recently successful in getting a $1 million grant from the Medical Research Future Fund, and they're on the pathway now, undergoing fundraising to really initiate a worldwide clinical trial in some of the key markets, with the aim to go into commercialization soon after. So I'll finish up there. I'll just give you, there's the website for Bionic Vision Technologies, where you can find out more about the commercialization. So thank you for your attention. Also the funding that we've received from the Australian government through the Australian Research Council. And the research teams, too many people, too numerous here to be to list all of them. We had up to 260 people involved in the Bionic Vision Australia research program, and enormous amount of support from all of the partners and members of the research consortium, to whom we're all indebted for this wonderful work that I've been able to tell you about today. So thank you very much for your attention.
Thank you so much, Tony, for sharing with us your incredible work over such a long period of time. We're going to hold off on questions until the end, and it's wonderful to see a few flooding into the Q&A chat. So please keep those questions coming. We will get to them right at the end. I'd now like to introduce our next speaker for the evening, Brendan Fafiani. He's the Chief Executive Officer of Cyban. Brendan is recognised as a medical technology expert with global experience in medical device product development, operations, quality management, regulatory affairs, manufacturing and supply. Brendan has successfully developed, achieved regulatory approval, launched and enhanced a range of medical devices from software to auto-injectors and diagnostics in startups, as well as in large commercial organisations. Brendan is currently the CEO of Cyban, which is focused on commercialising a non-invasive brain pulse oximeter. Thank you so much for joining us, Brendan.
Thank you, Sandra, for that very kind introduction, and thank you for the invitation this evening. A really fascinating presentation, Tony, and a really fantastic evolving story. So thank you for sharing. And it was really refreshing to hear you talk about those clear commercial goals right at the start of the project, because it ties in quite nicely with what I plan to cover this evening. Despite that fantastic introduction, and before I move on to the main topic, I thought it might be appropriate to give a little bit more of an overview of my background, and particularly around the commercializing of medical devices. So I've worked in life sciences right from the start of my career, despite a my screen is showing, sorry. Despite a very short stint with Japanese tobacco, which was a very different experience, to say the least. But for the last 12 years or so, I've been focused exclusively on medical device development. Whilst I was at Merck, which is a large German pharmaceutical company, I was responsible for launching a number of softwares and medical device products. These were mainly targeting the diabetes and the growth hormone deficiency space. And later on, taking on the responsibility for the lifecycle management of their rather large device portfolio globally. And these were mainly complementary drug delivery devices. More recently, and probably more appropriate to this evening, was the work I did with Global Kinetics. This is a company that was at the Florey Institute here in Melbourne. The founders developed a novel method of quantifying the symptoms of Parkinson's disease, and then providing a report to clinicians to guide their treatment decisions. So we launched the PKG, that was the Parkinson's kinetigraph, into 18 countries. And by the time I left the company in 2019, the PKG had been used by more than 50,000 patients. And it still remains the only commercially available and objective measure for Parkinson's today. I joined Cyban in 2020 as a CEO, and again, it's another innovation that has come out of Melbourne, created by a very talented physician based out of St. Vincent's Hospital. Our technology is set to disrupt how acute brain injuries are managed. Our brain pulse oximeter provides a non-invasive, reliable measurement that's able to detect a cerebral complication sooner than the current standard of care, which is at the moment requiring a neurosurgical procedure to place an invasive intracranial pressure probe or an oxygen probe directly into the patient's brain. So for us, it's quite a compelling value proposition to measure somebody's brain oxygen levels using near-infrared light rather than drilling a hole in the patient's skull. Of course, so it's articulating actually in detail a value proposition like this and how this is relevant to an innovator as they present themselves to investors that I was going to talk about this evening. It's cliche, but it really is a marathon and not a sprint. I think the rule of thumb is that it takes approximately 17 years and $50 million from revenue to first sales. Administrators in the healthcare systems are always making choices about what to spend within a capacity constrained system. So as entrepreneurs, we always need to keep in mind, no matter how amazing our innovation might be, that to bring it to market and ultimately have an impact on patient outcomes, we really need to be successful in changing behavior within the clinic and in convincing healthcare administrators to divert funds from their existing products and services to our products and services. So to consistently be able to raise capital during that marathon, the pitch always needs to be focused on aspects of creating shareholder value and not just the innovation itself. So to illustrate that, we use a very simple equation at Cyban. Shareholder value equals innovation plus customer value plus the business model. So customer value can be defined as a product or service that helps a target customer do more effectively, more conveniently, and more affordably a job that they have been trying to do. Where a customer job is a job that arises in the customer's life for which a product is being hired. And it's important to note that it's the job and not the customer that should be the fundamental unit of analysis when communicating with investors. And lastly, of course, the business model is made up of the resources, the processes, and the profit formula that you need to deliver on that customer value. So the key question becomes, how do we create and deliver customer value in a way that is sustainably profitable? I think in Australia, we're very good at managing technical risks. We develop great technologies, but we can often be poor at managing commercial risks, especially early on. There's a relatively long list of medtech companies who've raised significant funds, but have failed to gain broad adoption in the use of their device, and as a result, have failed to gain a return on invested capital because of a lack of thinking about what needs to be incorporated into those products quite early on. Of course, the journey all starts with the innovator, but to be set up well with investors, it's important to be able to demonstrate that you have a really intimate knowledge of the jobs that need to be done within that particular domain. Ideally, they'd like to see some indicators of success and some validation of the technology quite early, be it the awarding of grants, publications, or some quality research papers. Some base level of evidence generated either from a proof of principle or a proof of concept device, with at least some level of IP protection and some level of funding in place, whether it be self-funded or through an accelerator program, MedTech's Got Talent, something to that effect. The next common hurdle is ensuring that the innovator is willing to undertake what is often very targeted translational research, not just continuing discovery and exploratory research. I think it's important to accept that the innovation itself is only one component of success. And from this point, it's no longer just about the research. So understanding that commercializing the innovation is going to be a very long journey, and one that requires bringing on external expertise. So it's important that the innovator is willing to cede some control over the commercial decision making in the company. So what drives shareholder value? It starts with the credibility of the innovator, the innovation itself, and the IP that surrounds it. So the foundations of shareholder value are really the size of the market opportunity and the time and resources that it's going to be required to take that innovation to the market. And then the long-term success will be determined by whether you can establish that sustainable endorsed value of your therapy and your ability to commercialize it. So we tend to communicate the size of the prize as the unmet clinical need described within this jobs to be done framework. The relevant patient population, not just the prevalence and incidence of the broad patient group that your indication might be valid for. And also a realistic assessment of your comparators is essential. I think what is the existing gold standard? What is coming down the pipeline in development? And I think it's important to acknowledge that the answer to this and the way to describe this is never there are none. Often in medical devices, the comparator might be the clinician themselves. And I think this needs to be acknowledged quite early on because it creates its own unique set of challenges in being able to describe value. And lastly, the cost effectiveness of your therapy. What is the potential gain with your device that can be demonstrated in real world use? And I mentioned the jobs to be done a few times already. I would encourage you, if you're interested, to take a look at disruptive innovation and the jobs to be done framework. I think it's particularly well suited to novel and early stage medtech products. So time is your enemy. The more it costs and the longer it takes, the harder it is going to be to continue to fund an opportunity out of an Australian base. So there should always be a focus on wherever we can on reducing the time to the first market, but keeping in mind that creating sufficient high quality published evidence that really will influence change in behavior in the clinic and influence the purchase decisions takes a lot of time to generate. So starting that evidence generation early in your product development processes is really an important step. So this is where the real work begins in establishing that sustainable endorsed value of your therapy. It has essentially three foundational pieces, if you like. That's assessing the market access potential, undertaking your own health technology assessment, and then gaining endorsement from relevant stakeholders. Making an early assessment of your market access potential is a key first step, because using a really simple process of looking at where you might sit on a simple 2x2 matrix like this is a really helpful exercise. Obviously, being in the bottom right-hand box is ideal, but it's often not the case. So the key to getting value out of an exercise like this is having an early hypothesis of how you will define benefit of your device and for whom. So you can easily communicate this, and I think this will really help in increasing the likelihood of success of the project. Undertaking your own health technology assessment, as I said before, is essential. I think building up your own value dossier from day one and using hypothesis about the outcomes and the value, and then continuously testing these throughout your product development process, even with quick and dirty trials, is the way to build those foundations of sustainable endorsed value. And at that point, we would suggest mapping your relevant stakeholder groups, engaging with them as early as possible in your product development process to test those hypotheses about clinical need and customer value. and then using that stakeholder group to help guide your evidence generation strategy, or your long-term evidence generation strategy. I think these are all essential activities that help reduce the time taken and the costly missteps along the way of providing that sustainable endorsed value. So once all of that foundation work is done, we can start thinking about which countries to launch into. You can, again, use a fairly simple framework to help guide the decisions. Are the clinical guidelines supporting the use of your therapy in the country you're looking to launch into? Is there a clear payment pathway, or does one exist? What is the size of your relevant patient population? And is your therapy going to have a broad public health policy support? I think once you understand those, then you can determine whether you have the capability from within Australia to execute on your market entry plan. Who do you need to bring on, and how are you going to fund that? Before I wrap up, I did have to give credit to Andrew Maxwell from Chatsworth Associates for a lot of the content in the presentation today. I've worked with Andrew on a number of occasions over the past several years, and I'd really encourage you to reach out with him if anything, any of the content today resonated with you, or if you need some guidance or support on your own commercialization journey. So if you think about some of the aspects I described within the presentation today, I think collectively they can actually be used as a basis of an investor presentation. I think this can really help a lot in not only raising capital, but bringing in the right investors, or the right investor or investors that are able to assess the risk appropriately and assist in funding you over a relatively long journey that's going to be full of ups and downs, and really give the best opportunity to be successful in creating that shareholder value. So that pretty much wraps it up. It was quite a short presentation, but do feel free to reach out to me over email or LinkedIn. I'm more than happy to take any questions you might have. I might not have the answers, but I'll certainly be happy to put you in touch with people offline who might be able to help.
Thank you, Brendan, and you've brought us back neatly on time. So that does allow a few minutes for questions. There's some questions being posted, which I think Brendan and Tony can see. But let me ask the question that Doreen's posted first, because she's directed to both of you. So perhaps, Tony, you've had a minute to think about it. So can you perhaps talk about the team that is needed to harness to go from the science to a successful device, and then we'll get Brendan's opinion on that question as well.
Yeah, certainly. So we were guided, we actually gave this a lot of thought when we were setting the whole team up, and we were guided very much around what happened with the development of the cochlear implant, because that's been sort of one of Australia's greatest successes. And talking to Professor Graham Clark, I mean, one of the things he did was to bring all of the people, the different components of the technology, to bring people together at a very early stage to make sure that there was input from all of those people right through from the early stage design right through to the clinical implementation. So having, like, for example, in our program, having the surgeons on board right at the design stages of the technology was absolutely crucial. So it was really mapping out exactly what needed to be done, finding out where that expertise lay, bringing those people into the program, and making sure that they were talking with each other and working together on a common goal right from the very outset was absolutely crucial.
And Brendan, would you like to also comment on that?
Sorry, Sandra, I've just popped up the Q&A screen. Would you mind repeating the question?
So it's the third question down. Can you talk about the team that's needed to harness to go from the science to a successful device?
Yeah, so I'm heavily biased, of course, but I think bringing in commercial expertise as early as you can to help guide your decisions is essential. And I'd really agree with a lot of the points that Tony made. I think now at Cyban, what we're trying to do is establish our own stakeholder network and our own set of key opinion leaders, which is really a group that helps us validate our plans quite early on. So I would say those are the two key areas, understanding that I think finding technical capability in relative terms is not so difficult. So.
And so let's lead on to the other question that's been posted for you. So do you feel that universities understand how to incentivize clinicians to take this journey into commercialization? How do you get them to make that transition away from discovery-based research?
I would say that's quite difficult. I think I would say it's hard to force based on my experience. I think the innovator needs to be willing to go on their journey, and often they've had a lot of time to get there. It's many years working on this. So I think, yeah, I don't have much experience working directly with universities, so I'll keep my answer on that one brief.
I don't know whether Tony wants to chip in on that one, but I'll ask Tony a different question, which has been posted. The first one, for the company that's been established, BVT, how long do you expect them to need to operate prior to any commercial revenues? I know that Brendan quoted 17 years. Is that realistic?
It depends on where you start the race from. So we started in 2010, and you know, I think for a lot of medical bionics devices, that's not an unreasonable sort of assessment. We the way that our thinking in terms of what we were doing with Bionic Vision Australia was planned was very much along looking at what happened with Cochlear. So they had a very small market share right at the very start. They're aiming at the people with most severe deafness, and then basically building their patient population with the evolving nature of the technology. So they were able to address more and more people. And that's really, I think, what we're aiming to do with the bionic eye. The moment is really those who have the most severe form of vision loss. But as people become more confident with the technology, the patient group will gradually expand and become available to more and more patients.
Thanks, Tony. There's a couple of other questions here, but they're quite specific. So I don't know whether it might be better for you to answer these offline, but there's a question about whether you can share the go-to-market plan envisaged for the bionic eye. I don't know whether you want to respond.
I'm not
to
actually
that
part of the corporate structure and BVT, like and not part of the actual company itself. So we actually do consulting for them, and particularly the development of the next generation devices beyond the ones that are actually being tested out in patients now. So I'm not privy to their plans for that.
Okay, and there's another question here from Michael. Oh, there's a couple coming in. There's one from Michael about whether he can actually recruit a couple of people in your trials. I'll let you perhaps answer that one by email. I can put you in contact with Michael if needed. And there's finally a question here that might be for Brendan. If commercialisation of any medical device can be considered a project, would it fall under more classical project management methodology such as waterfall, or can incorporate agile features as well? I don't really want to give a quick answer because we're running out of time. Brendan?
I think there's probably a few answers to that one, but considering that this is a very long journey, I'd have to say this industry lends itself more to a waterfall approach. Having said that, depending on your device, really, I've been successful in incorporating a more agile approach within a waterfall, particularly if your device has a large software component, which is a little bit more akin to that iterative development cycle and trying to get that value out the door quite quickly. But certainly from a regulatory perspective, most of the regulators are looking for that very sequential waterfall style of delivery before you move on to your next phase.
Thanks, Brendan. And we are out of time, so I'm going to throw back to Bronwyn to close the event. Thank you.
Thank you, Sandra. And thank you so much, Brendan and Tony, for sharing your insights, your strategies. And it's just been a truly inspiring hour. Thank you so much. We would ordinarily be giving you a bottle of red wine at this point in time, but we'll find a way to get one to you. I'd like to just thank everyone for attending and remind you that the ATSE Awards are taking place next Thursday evening on the 10th of June, and there'll be more information and a virtual link so that you can join in and celebrate the success of some of our rising stars. And as for the Victorian division, our next event is on the 1st of July, and we have a wonderful speaker, an absolute expert, Doug McFarlane from Monash University, who will talk to us on energy and energy storage. So let's hope that we can meet there in person, and thank you so much for attending this evening. I'll look forward to seeing you all soon. Thank you.